Hi, I'm Aurel Enriquez, and this video contains our discussion on Chapter 15 of Mahon's textbook of Diagnostic Microbiology, which talks about the Streptococcus species, Enterococci, and other gram-positive catalase-negative cocci. Here is the outline of our discussion. So let's first look into the general characteristics that is applicable to both Streptococci and Enterococci. Then let's find out how they are classified. So there are two ways, the Lansfield classification and the Smith and Brown classification. And then let's proceed to talk about the clinically significant species such as Streptococcus pyogenes, Streptococcus agalactiae, Streptococcus pneumoniae, and then let's look into the other groups like Group C, Group G, virulent streptococci, and then lastly, we'll talk about the streptococcus like organisms. Let's begin with their general characteristics. Some species, well, actually, a lot of them are part of the normal flora of humans. Most members of the genera Streptococcus and Enterococcus may behave like facultative anaerobes, but they're actually aerotolerant anaerobes. So, just a quick review, what do we mean when we say facultative anaerobes? They are capable of growing with or without oxygen, 'di ba? Parang optional lang, okay lang kahit walang oxygen, 'di ba? They would still grow, but they grow better in the presence of oxygen, 'di ba? That's for facultative anaerobes. But when we say aerotolerant anaerobes, what does that mean? They only tolerate the exposure to oxygen. No, the exposure to oxygen won't readily kill them, but they're also not capable of growing in the presence of oxygen, di ba po? So we would still have to move them to a different incubation setup wherein there is no oxygen source, di ba po? So again, that's for aerotolerant anaerobes. When we say aerotolerant, they only survive the presence of oxygen, but they can't grow in the presence of oxygen. So that's for aerotolerant anaerobes. But again, if we were to compare that with facultative anaerobes, they're capable of growing with or without oxygen, but growth is just better with oxygen. All right. So ayan po. Um, some species are capnophilic, which would mean that they require increased concentration of carbon dioxide, whereas growth of other species is stimulated by the increase of carbon dioxide concentration, but it is not required. Okay, ulitin po natin, sana naintindihan nyo po. There are some species that are capnophilic, which means that they require increased amounts of carbon dioxide, but there are some species that don't actually require carbon dioxide, but they will grow better in the presence of increased carbon dioxide. All right. So, ayan po. Again, that would vary from species to species. Microscopically, they are gram positive cells that are arranged in chains, just like this one or in this photo right here. Okay. So, macroscopically naman po, no? Macroscopically, their colony morphologies appear to be grayish, pinpoint, or small and translucent, as we can see right here in this example. All right. So the main descriptions that we need to remember is that they are gram-positive cocci. Anong arrangement? In chains. Okay. Next, kung colony morphology ang ino-observe, anong description yung kailangan natin tandaan? They are pinpoint and transparent or translucent. Sorry. Let's now look into the cell wall structure of both Streptococcus and Enterococcus species. They both have the typical gram-positive structure. So, i-expect po natin na makapal yung peptidoglycan layer, di ba po? But they also have teichoic acid and they also have group-specific carbohydrates, all right? They have these additional structures referred to as polysaccharides, except for the viridin strep. Okay, so mamaya po, when we talk about the viridin streptococci, just know that although ang viridin strep is meron sila nung typical na gram-positive structure, they don't have the polysaccharides that we're talking about in this slide. All right, so ayan po. Now, let's find out, ano ba ang significance ng polysaccharide structures na ito? These polysaccharide structures are the basis for the Lancefield classification. So the Lancefield classification is based on a serologic method that depends on the polysaccharide antigens that are present on these streptococcal cell walls. So this is also the most commonly used method of classification for Streptococcus species and other similar organisms. So yung flow po ng discussion natin will follow the Lancefield classification. So this was kind of like developed by Rebecca Lansfield in the 1930s, and again, it is based on the extracted carbohydrate or yung polysaccharide antigens on the streptococcal cell wall. All right. So group A, Streptococci, includes Streptococcus pyogenes and also its other strains and serotypes. Group B includes Streptococcus agalactiae, of course, including all its other strains and serotypes. Group C includes Streptococcus dysgalactiae and equi. Group D includes Streptococcus bovis plus the Enterococcus species. And ayun nga po, no, here in the Lancefield grouping, Streptococcus pneumoniae doesn't belong to any group. Let's look at this one right here. Okay, so wala pong Lancefield grouping ang Streptococcus pneumoniae. Okay, and lastly, we have the virulent Streptococci. I've mentioned this earlier, no? So yung iba po dito, like the anginosus group, may also be referred to as the group G Streptococci. All right, ulitin natin. Group A, Streptococci, Streptococcus pyogenes. Group B, Streptococci, Streptococcus agalactiae. Group C would include Dysgalactiae and equi. Group D would include Streptococcus bovis and yung mga Enterococcus. Next naman po, Group G, yung Angionosus group, Mutans, Mites, and Salivarius group. Okay, so sila po yung Group G Streptococci. Sino walang classification? Sino hindi kasali? It's Streptococcus pneumoniae. All right, and also yung viridans strep. All right, keep in mind the viridans streptococci is also not included in the Lancefield classification. Next is the Smith and Brown classification, which is based on their hemolytic patterns. All right. So you should review your chapter 8 discussion po. Okay. So dun po pinag-usapan natin yung iba't ibang mga hemolytic patterns. All right. And how do we include them in the description of the colony morphology. All right. Again, that's on chapter 8. But eto po, reviewin ulit natin to dito sa chapter 15. No? So let's now talk about the Smith and Brown classification, which again, it's based on hemolytic patterns. All right. So if you would remember from our chapter 8 discussion, hemolysis is the destruction of RBCs in their culture media, diba? Either on PAP or on SBA. All right. So if a bacteria is alpha hemolytic, this means that they are only capable of partial or incomplete hemolysis of the RBCs on the blood culture or blood agar plate. All right. So this alpha hemolysis no, would produce a greenish discoloration around the colonies on PAP, as shown right here. So ulitin po natin, no, the bacteria was not capable of fully destroying the RBCs that's present in the culture media. So it was only capable of producing a greenish discoloration around the colonies. So the the most well-known example of a or an alpha hemolytic streptococcus species is Streptococcus pneumoniae, as we can see right here in this example. So nakikita natin no dun sa loob nung green na marker or green na box, 'di ba? The colonies around them no, or sa immediate surrounding nila or very close to the colonies. Ayun nga po, medyo nagbago yung itsura nung blood agar plate, 'di ba? And yan nga po, that's what we're seeing as a greenish discoloration, and again that only indicates partial hemolysis. So again, we refer to that as alpha hemolysis. All right. So again, do not forget our example on an alpha hemolytic organism. It's Streptococcus pneumoniae. If a bacteria is beta hemolytic, this means that they are capable of complete hemolysis of the RBCs present in the medium. They were capable of completely or fully destroying the RBCs that is present on BAP or SBA. All right. So beta hemolysis would produce a clear zone around the colonies and ganito po yung itsura nila. Okay, so notice no that there is a very obvious change on the immediate surrounding of the bacteria, di ba? There's an obvious change dun sa culture media natin. Hindi na siya red, di ba? There's this kind of like clear halo or clear zone around the colony. All right. So again, when we see something like this, then this is referred to as beta hemolysis. So for this chapter, our examples of beta hemolytic streptococci would include Streptococcus pyogenes, and Streptococcus agalactiae. Alright? So don't forget that. Okay? Mahalaga po na tinatandaan natin yung mga examples given here. So again, beta hemolytic Streptococcus species would include Streptococcus pyogenes and Streptococcus agalactiae. Up next is, ayan po, up next is gamma hemolysis. So if a bacteria is gamma hemolytic, this simply means that they are non-hemolytic. All right, so they can't destroy, they can't lyse the RBCs in the culture media, not even partially, all right? So we rarely use the term gamma, all right? Kadalasan po, we just say that the organism is non-hemolytic, all right? Maybe you could use the term gamma in reporting, no? When writing it down. Kasi mas madali nga namang isulat yung symbol ng gamma. But when you're talking to one another, you could just simply say that the organism is non-hemolytic, all right? So as you can see, right here, no, there has been no change in the immediate surrounding of the bacterial colonies. Walang nagbago kasi wala naman siyang nasira na mga RBCs. All right, so they produce no change on the immediate surrounding of each colony because they were not able to destroy the RBCs. And an example of a non-hemolytic streptococcus species is Streptococcus bovis. Okay, now that we know the general characteristics of streptococci and how they are classified, let's discuss each clinically significant species. Streptococcus pyogenes, or the group A Streptococci, has been a known colonizer of the skin and throat of humans, making these sites the primary source of transmission, either by inhalation of contaminated droplets in case of pharyngitis, or pwede rin po yung skin contact in cases of pyodermal infections. Okay, so you have to correlate po, no, with regards to where these organisms could be found, correlated din po doon how they could be transmitted, diba? So again, if they are found on the skin, then transmission could include skin contact. If they are found on the throat or upper respiratory tract, then transmission would include inhalation. All right. So their colony morphology on BAP appear to be small, translucent, and beta hemolytic, as shown here on this photo to the left. So as we can see right here, now observe po natin no, that there has been complete clearing or clear zone or halos surrounding each colonies, di ba po? And another way that we could observe this better is by performing transillumination. So when we say transillumination, you take a light source and you put the back or the bottom of the culture media against that light source. So kung beta hemolytic po yung organism na ino-observe nyo, you should be able to see na the light would easily pass through the culture media kasi nga po, the bacteria was able to clear out the RBCs in their surrounding. All right. So again, in this example, ayan po, we're seeing a photo of a beta hemolytic Streptococcus species. All right. So ayan po. Next, ayan. Again, their their colony morphology has been described to be small or pinpoint, diba? If you would remember from the previous slide, small or pinpoint. They are translucent and they are beta hemolytic. So notice that they don't produce any pigment, diba po? Let's now look into the virulence factors of Streptococcus pyogenes. The following virulence factors are similar in a way that they are all structural, all right? If we would look at this illustration right here, so itong mga susunod na virulence factors po na i-discuss ko, sila ay structural. Ang ibig sabihin po, they are not something that the bacteria produces, rather, naka-incorporate na po itong mga to dun sa mismong structure ng cell walls nila. Okay? So again, when we say that something is structural, no, it exists within the already made structure or already developed structure of the organism. So these virulence factors, hindi na sila kailangang i-produce pa ng organism dahil they are readily found on the cell wall of Streptococcus pyogenes. The M protein is attached to the peptidoglycan of the cell wall and it extends to the surface. All right. So, di ba po, as a gram-positive bacteria, Streptococci also has a thick peptidoglycan layer. But Streptococci, no, for Streptococcus pyogenes specifically, what makes them special is that they have the M protein molecule that causes the streptococcal cell to resist phagocytosis and plays a role in adherence of the bacterial cell to the mucosal cells. All right? So, ayun po na naka-highlight na yung mga mahalaga nating kailangan tandaan. The M protein, aside from being anti-phagocytic, it also provides adherence. All right? So, there are more than 200 different serotypes and subtypes of the M protein. All right. But what we need to remember here is yung M1 serotype. All right. So the M1 serotype is mostly responsible for cases of pharyngitis, right? Next is the fibronectin binding protein or protein F, all right, which helps the bacteria attach better to the epithelial cells. Next, ang lipoteichoic acid naman po, it's also for adherence specifically to the respiratory epithelium. All right, so yun po yung similarities nitong tatlong ito, okay? All of them are incorporated onto the cell wall of the Streptococcus pyogenes, and all of them are capable of providing adherence. All right? Anong special sa M protein? Maliban sa adherence, it's also anti-phagocytic, di ba po? Next, for lipoteichoic acid, it's adherent pero specifically to where? To the respiratory epithelium. Up next is the hyaluronic acid capsule. All right. So as we can see right here, it's the same illustration, di ba po? It's also showing us that the hyaluronic acid capsule is also found no sa surface ng cell wall ng Streptococcus pyogenes. So this is also a structural virulence factor. All right. So the hyaluronic acid capsule prevents opsonization and phagocytosis by neutrophils or macrophages. This capsule allows the bacteria to mask its antigens and remain unrecognized by the host immune system. So kumbaga po, ito yung disguise nila, 'di ba? Tinatakpan nung hyaluronic acid capsule yung surface ng bacteria para matakpan din yung antigens para hindi sila ma-detect nung immune system natin, 'di ba? Because if you would remember the basic steps of phagocytosis, what's the first step in phagocytosis? Opsonization, 'di ba po? Yung mga antigens sa surface ng bacterial cell kailangan magbabind doon yung mga antibodies that has been produced by our immune system. But if the hyaluronic acid capsule is covering yung mga antigens ng bacteria, hindi sila mahahanap nung antibodies ng immune system natin, and the antibodies of our immune system would have nowhere to bind, 'di ba? And if they don't bind, phagocytosis doesn't happen. All right? So that's for the hyaluronic acid capsule. So again, this is one of the virulence factors of Streptococcus pyogenes. These next virulence factors are referred to as hemolysins. These are the ones responsible for the beta hemolytic activity of Streptococcus pyogenes. So first up is streptolysin O, or SLO for short. Okay, so this is an oxygen labile hemolysin. What do we mean when we say oxygen labile? It means that it could only produce hemolysis under anaerobic conditions. Okay, ulitin po natin, when we say that something is labile, its structure could be destroyed by exposure to a certain condition. So in this case, pag sinabi natin oxygen labile, the structure and function of streptolysin O could be affected if it is exposed to oxygen. All right. So again, we would only be able to observe its action anaerobically. All right. So either you incubate the culture or this bacteria anaerobically, or You could also observe this by stabbing the agar. All right. So this one is responsible for subsurface hemolysis. Okay. So what are we talking about? Ano pong tinitignan natin dito? When we say subsurface hemolysis, this is hemolysis at an area where they are no longer exposed to oxygen. Okay? So let's look at this plate right here. So ito po yung usual na four-quadrant streaking plate na ginagawa natin. Di ba po? So this is the first quadrant, second quadrant, third, and then this is the fourth quadrant. Di ba po? As you can see right here, to properly stab the agar and observe streptolysin O, subsurface hemolysis, babalik tayo papunta dito sa first quadrant, okay? And then using the same inoculating loop that you use to make the streaking, you create these little stabs dun sa first quadrant. So what's the rationale behind that? Why do you have to do that? Ang oxygen naman po, it does not have the capability to diffuse to the deeper parts of the agar. So if you inoculate a media by stabbing, yung mga mas malalim na areas po, hindi na ito abot ng oxygen. Okay? Thus, providing an anaerobic environment for the organism. Okay? So in this case, we were able to observe subsurface hemolysis dahil dito sa mga areas kung saan hindi na sila abot ng oxygen, they were still capable of showing beta hemolysis. Diba? So again, that is referred to as the subsurface hemolysis that it has been produced by streptolysin O. All right. So ayan po. Um, Now, by stabbing the agar, no, again, it creates an anaerobic environment. Kaya po siya tinawag na subsurface hemolysis. Now, if subsurface hemolysis is observed, kapag nakita nyo po na nagkaroon ng hemolysis in those areas where the stab was made, then this means that your streptococcal isolate can in fact produce streptolysin O and may possibly be Streptococcus pyogenes. All right. Take note that SLO is highly immunogenic, okay? Meaning our immune system can produce antibodies against this, all right? Yung streptolysin O daw po, kaya itong i-recognize ng immune system natin and our immune system could produce antibodies against streptolysin O. And ito po yung basis ng diagnostic test na anti-streptolysin O or ASO. So ASO, this is a test to determine whether a patient has had a recent infection with Streptococcus pyogenes. So if for example, previously nagkaroon ng strep pyo infection itong pasyente na ito, of course, we would be able to find out kasi its immune system was able to produce antibodies against SLO. All right. So again, this is to find out if a patient has had a recent infection with Streptococcus pyogenes. Again, that is the ASO titer. We aim to detect the antibodies produced against streptolysin O. Next is Streptolysin S. So kabaliktaran po ito ng SLO. Streptolysin S is oxygen stable. Okay? Streptolysin S, siya ay oxygen stable. Meaning, even after exposure to oxygen, it's still capable of producing hemolysis. Alright? Hindi po siya magiging apektado ng exposure to oxygen. So, if we were to look at this plate right here, simula po dito sa second hanggang third quadrant, na observe natin na even if we did not stab the agar, we were able to observe na beta hemolytic pa rin yung organism, di ba po? So again, that is because of the action of streptolysin S. Alright, so this is observed aerobically. Hindi mo na kailangang gumawa ng anaerobic environment. Okay? Now, this one is also non-antigenic. Okay? Again, kabaliktaran ito ng SLO. Ang streptolysin S po is non-antigenic, meaning kahit gaano karami pang streptolysin S ang i-produce ng isang organism, our immune system would not react to this. All right? Which is why we don't have an anti-streptolysin S titer, 'di ba? We don't have a test to detect the presence of streptolysin S kasi nga we won't be able to do that dahil hindi nga naman nagre-react ang immune system natin dito. All right? So that was for streptolysin O and streptolysin S. Streptokinase causes the lysis of fibrin clots by acting on plasminogen. So plasminogen together with streptokinase is converted to plasmin, which can cause lysis or destroy of fibrin clots. All right, so let's look at this diagram right here. So in our blood, no, sa ating plasma, mayroong plasminogen. Now, if an infection with Streptococcus pyogenes leads to the production of streptokinase. The streptokinase from the bacteria and plasminogen from our blood would combine and create a streptokinase plasminogen complex. Now, after this, after they have complex together, the plasminogen would eventually be converted to plasmin. All right, and plasmin upon or has the capability to act upon fibrin. All right. So fibrin is then degraded, leading to the lysis of fibrin clots. All right. Now, if you would remember from our discussion on the Staphylococcus species, we have a similar virulence factor. Di ba po? The staphylokinase. All right. Staphylokinase or fibrinolysin. All right. So they have the same function. All right, they're both capable of dissolving fibrin clots. The difference is that they're produced by different organisms. Okay, so ayan po. Again, streptokinase no is an organism or is a virulence factor that's capable of causing the lysis of fibrin clots by acting on plasminogen. Next are streptococcal pyrogenic toxins, formerly known as erythrogenic toxins. These are responsible for toxins that are formed during the cases of scarlet fever, which would lead to the production of rashes. Okay, so we'll talk more about that later. Pero kasama po sa mga prominent clinical manifestations ng scarlet fever ang erythematous rashes na meron yung pasyente. So there are four immunologically distinct or four serotypes of pyrogenic toxins that may be produced by strep pyogenes. And these are SPA, SPB, SPC, and SPF. All right. So these toxins function as super antigens and they could stimulate T lymphocyte proliferation, which leads to increased production of cytokines that can mediate the disease process. So, bale, dahil po sa mga pyrogenic toxins na no, nag-overreact yung immune system ng pasyente which leads to the production of other symptoms plus rashes. Okay? So that's for the streptococcal pyrogenic toxins. So we have the SPA, SPB, SPC, and SPF. All right? So again, no, I would like to remind you to always try to correlate depending on the virulence factor, ano po yung disease that we could associate with that. So again, with regards to these exotoxins or pyrogenic toxins, the disease associated with this is scarlet fever. Dahil nga po dito sa mga streptococcal pyrogenic toxins na to, na-observe natin yung characteristic rashes that is present among people with scarlet fever. Next, hyaluronidase is a virulence factor na kilala na rin natin, di ba? During our discussion on staphylococci. So hyaluronidase or the spreading factor is an enzyme that solubilizes hyaluronic acid. Okay? So this may then cause the separation of tissues and further spread of infection. Last one is DNase B. So DNase, this functions the same way as the DNase that is produced by Staphylococcus species. So again, it could break down the structure of DNA, leading to the lower viscosity of tissue fluids, and that would subsequently lead to further spread of the infection. Let's now talk about the infections and diseases associated with Streptococcus pyogenes. The following diseases are recognized as pyodermal infections. First is impetigo. This is a localized skin disease that begins as small vesicles that progress to weeping lesions. The lesions crust over after several days. Impetigo is usually seen in young children And this photo right here is an example of a patient suffering from impetigo. Next is cellulitis. So cellulitis can develop following deeper invasion by streptococci. There is erythema and edema or redness and swelling. So the infection can be serious or life-threatening if bacteremia or sepsis ensues. Okay? As we can see right here, no, tignan po natin itong pictures dito, or yung picture dito sa gitna, no. Um, Erythema and edema. There is redness and swelling, which is very hard to miss. It's super obvious po kapag may cellulitis yung pasyente. So if we look at this patient's leg right here, halos magka-same size na po yung calf ng pasyente and yung ankle nila, 'di ba? The calf and the ankles are almost the same size because of the intense edema. And of course, very apparent po ang swelling. So again, this is a clinical manifestation of cellulitis. All right. Up next is, ayan po, erysipelas. All right. So erysipelas, ayan, this is an acute infection of the skin and subcutaneous tissues where there is swollen and reddish spots or areas with irregular borders, as we can see right here in this photo. And this one is very common among elderly. All right. So again, erysipelas. This is characterized by an acute spreading skin lesion that's intensely erythematous and is not plainly demarcated, or it has a it's plainly demarcated, but it has an irregular edge, as we can see right here on this third photo. So, ayun po, no, again, tatandaan po natin yung population at risk nila. Okay? So, kapag sinabi natin it's more common among children, dapat ang maiisip yung disease ay impetigo. Pero kapag sinabi po na it primarily affects the elderly, dapat ang una nyong maisip ay erysipelas. Next is scarlet fever, or in some references, this is referred to as scarlatina. Infection with strains of Streptococcus pyogenes that produce the streptococcal pyrogenic exotoxins can result in scarlet fever. After an incubation period of 1 to 2 days, a diffuse red rash appears on the upper chest and spreads to the trunk and extremities of the patient. All right. So again, that would happen in the first 1 to 2 days. The rash disappears over the next 5 to 7 days and is followed by desquamation. Okay? So, medyo mawawala na daw po yung mga rashes, pero magkakaroon ng desquamation, meaning there may be some areas of the skin na natutuklap. All right? So, this photo on the left is an example of a patient suffering from scarlet fever. So, that's why it was referred to as scarlet fever, because the redness that was observed in the rashes of these patients, no, it has this certain shade of scarlet red. All right. So again, for the first 1 to 2 days, dun po natin ma-observe yung rashes, and then after 5 to 7 days, we'll eventually be able to observe desquamation. The mode of transmission of the scarlet fever is via inhalation of infectious respiratory droplets. Next is necrotizing fasciitis. Necrotizing fasciitis is an invasive infection characterized by rapidly progressing inflammation and necrosis of the skin, subcutaneous tissues, and fascia. This one goes way beneath the surface to the point that it can literally eat away the patient's flesh, which is why this has been referred to as the flesh eating disease. This is a life threatening disease, but death can be prevented if early intervention is done. The mortality rate may reach greater than 70% if the infection is left untreated. And in some cases, the entire limb is amputated. All right. So here's what you need to remember regarding the clinical manifestations of necrotizing fasciitis. Most of the time, no, the progression is very fast. Okay, within a few hours or within a few days, no, nagpo-progress po or nag expand yung infection. And usually, IV antibiotics, all right, intravenous antibiotics may not be enough to treat this infection. Dahil minsan po, mas mabilis na kumikilos yung infection kesa sa tumatalab yung antibiotics, all right? So madalas po, pinagsasabay na rin yung IV antibiotics, and debridement. All right. So when we say debridement, this is surgically removing the necrotic areas. Okay. So ayun po. Again, that's our keyword. Okay. Yun yung mahalagang term na kailangan natin tandaan. It's rapidly progressing. This usually results from trauma, physical trauma such as lacerations or burns. So yung mga burn victims po, no, they are at high risk of eventually developing necrotizing fasciitis. Now, depending on which organisms have been cultured when samples have been collected, necrotizing fasciitis may be characterized or, sorry, categorized as type 1, type 2, or type 3. Okay? So, a polymicrobial infection from which both aerobic and anaerobic bacteria have been recovered is categorized as type 1 NF. Okay, so NF stands for necrotizing fasciitis. Okay, so kapag multiple organisms daw po yung causative agent and it's a mix of both aerobes and anaerobes, then that is type 1. We have another form of type 1 necrotizing fasciitis, which is the saltwater NF. All right. So for saltwater NF, classified pa rin siya as type 1, pero iisa lang ang causative agent niya. So this is caused specifically by Vibrio vulnificus. We'll talk more about the Vibrio species when we get to chapter 20 of our discussion. But again, no, there are two subtypes of type 1 necrotizing fasciitis. First is polymicrobial. It's caused by both aerobes and anaerobes. Next, the second subtype of type 1 NF is the saltwater NF, which is specifically caused by Vibrio vulnificus. Next is type 2 necrotizing fasciitis. This one consists of only group A streptococci. Okay? So sa type 2 NF daw po, no, ang may kasalanan lang dito ay Streptococcus pyogenes. Right? Next is type 3 NF. Type 3 NF is otherwise referred to as gas gangrene or clostridial myonecrosis, which is caused specifically by Clostridium perfringens. So Clostridium perfringens po is a topic for another discussion. But ayun po, here are the three different categories of necrotizing fasciitis, and ang tatandaan natin dito is type 2 because type 2 necrotizing fasciitis is specifically caused by group A streptococci. Group A strep associated with streptococcal toxic shock syndrome produces a streptococcal pyrogenic exotoxin, notably SPEA. Okay, so these toxins likely play a major role in the pathogenesis of this disease, functioning as superantigens, leading to the overstimulation of the immune response, resulting in cytokine production or excessive cytokine production. So again, excessive cytokine production is bad. Tulad ng nabanggit ko po kanina, no? This is like an overreaction of the immune system, which could affect other organ systems and do more harm than good. Okay? So streptococcal TSS is a condition in which the entire organ system collapses, leading to death. The initial streptococcal infection is often severe, pero pwede rin po itong magsimula as pharyngitis, peritonitis, cellulitis, or wound infections, and the symptoms that develop are similar to the symptoms of staphylococcal toxic shock syndrome. So just a review, no? If you remember, in cases of staphylococcal toxic shock syndrome, these have been described to be menstruation related. All right? Ulitin po natin yun. Staphylococcal toxic shock syndrome has been described to be menstruation related. All right. So usually po, when the bacteria is present and it has contaminated a person's tampon, no, it's a period product for people watching, no. Imagine a tampon and then it has been contaminated with Staphylococcus aureus, and then it is used by this specific person. This person now has a risk of developing staphylococcal toxic shock syndrome because when the bacteria produces their toxins, maa-absorb po ito dun sa mucosal epithelium nung person using the tampon, leading to, ayun nga po, no, yung menstruation-related toxic shock syndrome. That's for Staphylococcus aureus, okay? But if we're talking about streptococcal toxic shock syndrome, dito naman po, a person needs to have had a previous infection with Streptococcus pyogenes before the toxic shock syndrome happens. All right. I hope you understood no the difference. Again, for Staphylococcus aureus, this leads to menstruation related toxic shock syndrome. All right. Again, that's Staphylococcus aureus. But if we're talking about streptococcal toxic shock syndrome, what happens here is nagkaroon muna po ng initial infection or ibang type of disease yung pasyente that was caused by Streptococcus pyogenes, which eventually led to the toxic shock syndrome. All right. So again, as we can see right here, initial infections would include pharyngitis, cellulitis, or wound infections. And this could also be observed in patients that are often bacteremic or have NF. All right. Maaari din po na kapag may necrotizing fasciitis yung pasyente, mag-progress ito papuntang streptococcal toxic shock syndrome. All right. So again, do not forget, as you can see on the final bullet or on the last bullet, this this disease no is attributed to the production of the pyrogenic exotoxin SPA. Most common clinical manifestations of Streptococcus pyogenes are bacterial pharyngitis, tonsillitis, which is commonly referred to as strep throat. Most cases of strep throat may also be caused by Streptococcus species from other groups, particularly Group C and Group G. They also have the capability to produce significant acute pharyngitis, but this is less commonly seen. Okay, ulitin po natin yon. Strep throat is commonly caused by Streptococcus pyogenes. But there are other Streptococcus species from group C and group G that could also cause strep throat, although that's quite rare. Okay? So kapag tinanong kayo, what is the causative agent of strep throat, mas pipiliin nyo pa rin po dapat isagot ang Streptococcus pyogenes. So after an incubation period of 1 to 4 days, an abrupt onset of illness would proceed. So this would include sore throat, malaise, fever, and a headache. So the tonsils and pharynx are inflamed and the cervical lymph nodes are swollen and tender. Okay? So upon physical examination with the doctor, possible po na makakapa ng doctor yung lymph nodes nung pasyente dahil sa pamamaga. Alright? Minsan po, there may also be symptoms beyond the respiratory tract such as nausea, vomiting, and abdominal pain. Okay, ulitin po natin. Minsan, no, the symptoms of strep throat doesn't stay respiratory. Some patients may also experience nausea, vomiting, and abdominal pain. So these are the disease. No, the disease ranges in intensity, and in some cases, symptoms may not be seen. So iba-iba rin po yung disease manifestation per patient. Okay, so pwedeng yung iba, mild symptoms lang, while the others could present with all of those symptoms previously mentioned. Okay? So the symptoms usually subside within 3 to 5 days unless complications such as peritonsillar abscesses would occur. Okay, so again, this is referred to as strep throat. Acute rheumatic fever typically follows strep pyogenes pharyngitis. Ulitin ko po, acute rheumatic fever develops among people who have had repeated infections of strep throat. This is characterized by fever and inflammation of the heart, joints, and blood vessels, and also subcutaneous tissues. Symptoms usually begin within 1 month after the infection. Most serious result is chronic progressive damage to the heart valves. All right. So, repeated infections with strep throat can produce further valve damage. Kadalasan ito po yung nagiging consequence para dun sa mga pasyenteng paulit-ulit yung pagkakaroon nila ng strep throat. The pathogenesis of rheumatic fever is due to the antigenic cross-reactivity between streptococcal antigens and heart tissues. Okay, ulitin po natin yun. May antigenic cross-reactivity daw po ang streptococcal antigens at yung heart tissues of humans. All right, so there are structures on the surface of Streptococcus pyogenes that is similar in structure with our cardiac tissues. Kaya po, minsan yung mga antibodies, no, ang problema po dito, merong similarity yung structure nila. So, yung mga excess na antibodies na produce during the course of infection with strep throat, they eventually recognize yung cardiac tissues as the same antigens to be acted upon. So yung mga antibodies, dun tuloy sila magbabind sa cardiac tissues. Okay? So ngayon, aakalain ng immune system na kalaban din yung cardiac tissues. Kasi bakit nga naman dun kumapit yung antibodies, 'di ba? But again, they were only there just because of antigenic cross-reactivity, just because there are similarities with the antigenic structure of Streptococcus species and the structure on the surface of heart valves. All right? So magkahawig lang po sila, just to simplify, no? So ayan po, this would lead to tissue damage because now the immune system is acting upon the heart valves where the antibodies have binded. All right? Another thing that we need to remember is Sydenham chorea. So Sydenham chorea, this happens when antibodies also cross-react with brain tissues, leading to neurologic manifestations. Okay, so hindi na lang sa heart valves pwedeng mangyari ito. Sometimes cross-reactivity could also happen with brain tissues. And when that happens, this would now be referred to as Sydenham chorea. But again, the pathogenesis is the same. Okay, there is excess antibodies. These excess antibodies bind to structures that are similar or cross-reactive with streptococcal antigens. Pero ayun nga po, pag acute rheumatic fever, ang main target dito ay yung heart valves. Pero kapag Sydenham chorea, ang main target dito ay brain tissues. Up next is acute glomerulonephritis or AGN. Acute glomerulonephritis may occur after either a cutaneous or pharyngeal infection. Okay? So dito po, kahit hindi pa ulit-ulit, no, basta nagkaroon ng isang person ng cutaneous infection with strep pyogenes. What are examples of cutaneous infections by strep pyogenes? Impetigo, cellulitis, and erysipelas, 'di ba po? Nabanggit natin kanina. So, um, the pathogenesis appears to be immunologically mediated, all right? Nagkakagulo na naman yung immune system dito. So, circulating immune complexes are found in the serum of patients with acute glomerulonephritis. And this is thought that those antigen antibody complexes deposit in the glomeruli. Okay, ulitin po natin, 'di ba? Yung Streptococcus pyogenes may antigens sa cell walls. Next, yung immune system natin magpo-produce ng antibodies. So they bind together creating these antigen antibody complexes. All right? So what happens, no? These antigen antibody complexes, minsan po, they would go through our circulation. And di ba po, one of the functions of the human kidneys is to filter the blood, di ba? Again, if may mga natira po sa bloodstream ng pasyente na antigen antibody complexes from a previous infection, these may be deposited or get stuck in the glomeruli as blood passes through the kidneys. Because if you would remember from your anatomy classes, di ba, yung glomerulus naman po natin, they're only capable of filtering small particles, or very small particles. Now, for large particles such as proteins, hindi na po ito kayang padaanin ng glomeruli, 'di ba po? So usually these antibodies and antigen complexes, since they are made up of proteins, they are too large to pass through the glomerulus. So again, naiipon lang po sila doon sa kidneys nung pasyente. So since napansin nung immune system na andun sa kidneys naiipon yung antibodies, iisipin na naman nung immune system na andun din yung kalaban, which would lead to the immune system attacking the kidneys, which would lead to renal impairment of kidney function, all right? Or impairment of kidney function. Okay, so ayun po. I hope you understood, no, how acute glomerulonephritis happens. So minsan nako-confuse, na minsan nako-confuse yung immune system natin, 'di ba? Na minsan nasa maling lugar magbabind yung antibodies, minsan may ipon sila sa maling lugar, so dun sila ngayon, kumbaga, dun magwo-work yung immune system natin, alright? Where it shouldn't be, diba? That shouldn't happen. But again, it's quite unavoidable with all these antibodies floating around after a previous infection. Anyway, both acute rheumatic fever and acute glomerulonephritis are referred to as post-streptococcal sequelae. Okay, so when we say post-streptococcal sequelae, this means that they develop after a previous streptococcal infection. Again, when we say post-streptococcal sequelae, this means that these are diseases that develop after a previous streptococcal infection. Meron munang naunang streptococcus infection yung pasyente that led to the development of these two diseases. Let's proceed to laboratory diagnosis. First up is Taxo A or bacitracin susceptibility. This would help us differentiate strep pyogenes from all other beta hemolytic streptococci, specifically from throat cultures. So, with an antimicrobial disc containing 0.04 units of bacitracin, if the isolate is a group A streptococci, then this should give a susceptible result. All right, so magkakaroon po ng zone of inhibition around the disc as we can see right here. Sa chapter 12 and 13 pa po natin pag-uusapan yung antimicrobial susceptibility testing, no? But as we can see right here, when we say zone of inhibition, it's a specific area where the bacteria was not capable of growing. All right, so i-strip mo yung bacteria dun sa plate, no, inoculate mo yung plate, then tsaka mo ilalagay yung antimicrobial disc. So kapag tumubo siya everywhere else except in the immediate surrounding of the antimicrobial disc, no, it means that the bacteria is susceptible. This could depend on measurements as well. May measurements pa po yung mga zone of inhibition na yan. Okay, but for now, no, just to simplify, if the organism had a zone of inhibition, then it could signify that this is susceptible to bacitracin. All right? So, ayan po, this is referred to as taxo A. Bakit taxo A? Because it's supposed to identify group A streptococci. Next is PYR or pyrrolidonyl arylamidase test. This is actually more specific than Taxo A because there are other similar organisms out there that may also be susceptible to bacitracin. So again, if you would like to be more specific, you could perform pyrrolidonyl arylamidase. So if you would remember from our discussion on Streptococcus species, di ba napag-usapan na rin po natin to? Pyrrolidonyl arylamidase could help differentiate Staphylococcus aureus from Staphylococcus lugdunensis, 'di ba po? Pero pwede rin natin 'tong gamitin for the Streptococcus species. All right? So again, in this case, we could use it to identify Streptococcus pyogenes and differentiate it from all the other beta hemolytic streptococci. So this detects the presence of the PYR enzyme produced by strep pyogenes. And a positive result is shown here on the left. This is indicated by a production of a cherry red color after detecting or after adding the detector reagent. This slide shows us a list of all the other additional tests that we could perform, but do take note that dito sa lahat ng to, Streptococcus pyogenes would test negative, all right? Because there are other organisms out there that would test positive for these, all right? So let's proceed to the others. Next is the group B Streptococci or Streptococcus agalactiae. So all strains of Streptococcus agalactiae have the group B specific antigen, which is why sa Lancefield grouping, sila po yung group B Streptococci. So they may be found as part of the normal flora of the lower gastrointestinal tract and the genital tract. Okay? So its primary virulence factor is its capsule, composed of sialic acid. This capsule prevents phagocytosis but becomes ineffective after opsonization. All right? So take note of that, no? May polysaccharide capsule po ang Streptococcus agalactiae with the addition of sialic acid. But this does not mean na safe na sa opsonization ang Streptococcus agalactiae. All right? Opsonization could still happen even with a capsule for Streptococcus agalactiae, and then this could inactivate or make the capsule ineffective. All right? So ayan po. Although nakakatulong po yung capsule niya in avoiding phagocytosis by masking its surface antigens, if they encounter any free antibodies that ends up covering them or opsonizing them, wala din po. No, opsonization would render the capsule useless. Okay? So, strep agalactiae has been associated in many different nosocomial infections, but the most notable disease is neonatal meningitis. All right. So, colonization of the vaginal and rectal area with group B strep is found in about 10 to 30% of pregnant women. So, dun po nakukuha ng mga neonates yung infection. All right. So, if for example, the vagina um, has been infected with Streptococcus agalactiae and then the baby passes through the infected birth canal, doon niya po maaaring ma-acquire yung bacterial cells that could lead to the infection. All right? So most infections of infants occur in the first 3 days after birth and usually within 24 hours. All right? So again, the most notable disease that is associated with Streptococcus agalactiae is neonatal meningitis. Now, this is from our former review professor during our time. No? Paano daw natin matatandaan na ang group B streptococci ay nagdudulot ng neonatal meningitis? Neonates are babies, 'di ba? So, group B, strep. Meningitis for babies. All right. So, ayun po. If you have better ways to remember that, that's cool. But if you don't, ayun po. No? Group B streptococci, it causes meningitis for babies. At ang tawag sa infection na yun ay neonatal meningitis. Group B strep colonies on BUP or SBA are grayish white and mucoid, which are surrounded by a very small zone of beta hemolysis, as shown here on figure 15.5. Upon gram staining, we'll be able to observe gram-positive cocci in short chains kung diretso galing sa clinical sample yung ig-gram stain mo. But longer chains could be observed if the colonies were taken from culture, as shown here in these photos. Next is the CAMP test or the Christie Atkins Munch Peterson test. This can differentiate strep agalactiae from other beta hemolytic strep. Okay? So ulitin natin kanina, we have the group A streptococci, di ba? Yung strep pyogenes, which is also beta hemolytic. What are the tests that's going to help us differentiate strep pyogenes from all the other beta hemolytic strep? It's the Taxo A at saka PYR, 'di ba? Now, in this case, for strep agalactiae, what test is going to help us differentiate it from all the other beta hemolytic strep? It's the CAMP test. All right. So we could either use a beta lysine strip. Okay. So it's a kind of like a paper strip that has been filled with beta lysine, or we could also use an organism that produces beta lysine, such as Staphylococcus aureus. Okay. So the principle behind this is that certain bacteria, such as group B strep, can produce an extracellular hemolytic factor known as the CAMP factor. Alright? So ulitin po natin, no? Ang Staphylococcus aureus, for example, kaya niyang mag-produce ng beta lysine. Pero ang Strep agalactiae naman po, ang kaya niyang i-produce ay camp factor. All right. So this camp factor can act synergistically with the beta lysine that has been produced by Staph aureus, causing an enhanced hemolysis of the RBCs in the medium. So what we're going to do here is that we'll streak the suspected group B strep isolate perpendicularly to the Staphylococcus aureus streak. All right. So a positive reaction is indicated by a or an arrowhead zone of hemolysis that is adjacent to the area where the two streak lines would meet, as we can see right here in this example. Okay, so ulitin po natin, yung beta lysine na pinoproduce ng Staph aureus plus yung CAMP factor na pinoproduce ng Streptococcus agalactiae equals enhanced hemolysis. And again, what is the specific shape of this hemolysis? Bow tie or arrowhead. So parang pa-triangle po siya, as we can see right here in this example. So this one shows us a positive CAMP test. Purate hydrolysis is also a very useful test to differentiate strep agalactiae from other beta hemolytic strep. If the bacteria produces the enzyme hippuricase, then this can hydrolyze hippuric acid. Hippuric acid would become glycine and benzoic acid. Ngayon, yung glycine po na na-produce through that reaction, this can be detected after the addition of ninhydrine, forming a purple colored product as we can see right here in this picture. So, ang positive result po is a purple color. But again, you would only be able to observe the purple color after adding ninhydrin. Proceeding with the next group, Group C and G Streptococci, which are subdivided into large colony and small colony forms. Large colony forming beta hemolytic isolates belong to Streptococcus dysgalactiae, subspecies Equisimilis. Meanwhile, the small colony forming beta hemolytic isolates belong to the Streptococcus angionosus group, which is also included as a virulent streptococci, but we'll talk more about that later. Clinical infections by Streptococcus dysgalactiae subspecies Equisimilis, although uncommon, they have involved several body sites. The spectrum of infections resembles that of Streptococcus pyogenes, and this includes upper respiratory tract infections, skin infections, soft tissue infections, and invasive infections such as necrotizing fasciitis. All right, so ulitin po natin yung mga diseases na kayang idulot ng Group C and Group G. Streptococci is very similar to those that could be caused by Group A, Streptococci or yung Streptococcus pyogenes. It's just that it's not very common that Group C and G would cause infections. Kaya mas kilala pa rin po natin ang Streptococcus pyogenes na nagdudulot ng mga diseases na nasa listahan na ito. Next is Streptococcus pneumoniae. S. pneumoniae is actually a member of the Streptococcus mitis group, which is a virulent strep. But this one is much more virulent than all the other members. Strep pneumoniae can express one of approximately 90 different capsular types based on chemical variations of the capsular polysaccharide. So that is strep pneumo's primary virulence factor. Okay, ulitin po natin, the primary virulence factor of Streptococcus pneumoniae is its polysaccharide capsule. The antibodies directed against the capsular antigen is protective, meaning immunogenic po yung capsule ng strep pneumo. So our immune system can recognize this and make antibodies against it. Upon microscopic examination, we should be able to see gram-positive oval or lancet-shaped cocci in pairs, as you can see right here in this picture. So notice po, di ba, that they're not perfectly spherical, no? Medyo parang pa-oval po yung shape nila. So they're characteristically described as lancet shape. Also, madalas po sila nakikita Bitus. Okay, so they're usually referred to as pneumococci. Streptococcus pneumoniae is well known as the causative agent of lobar pneumonia. The bacterial cells, once they reach and colonize the alveoli, they can stimulate an outpouring of liquid, which serves to facilitate the spread of the organism to adjacent alveoli. So magkakaroon po ng tubig sa baga or accumulation of fluid in the patient's lungs. That process stops when the fluid reaches the fibrous septa that separates the major lobes of the lungs. All right? So kaya po siya tinawag na lobar pneumonia. They are confined to a specific lobe of the lungs. Now, if you would remember from your anatomy classes, diba, if we could see right here in this illustration, our right lung is divided into three lobes. Meanwhile, our left lung is divided into two lobes. So as we can see right here, yung first lobe or yung nasa taas, This is what it looks like if the alveoli is not infected, diba? So, and po, there's airspace, no, gas exchange would happen naturally. But let's compare that to the lower lobe of the patient in this illustration, wherein dito nga po natin ma-observe yung lobar pneumonia. Naka-confine lang siya dun sa specific lobe. But as you can see right here, there has been fluid accumulation dun sa airspace sana nung alveoli. Okay? So again, the spreading of the infection would stop when the fluid that has accumulated reaches the fibrous septa that separates the major lobe of the lungs. Kaya po, confined lang siya sa isang lobe. So, this accounts for the lobar distribution of the infection, hence the name lobar pneumonia. So, as shown here in this illustration, ayun nga po yung kumbaga parang visual representation ng disease na yun para maintindihan natin kung ano ba ang ibig sabihin ng lobar pneumonia. In children, strep pneumo may also cause otitis media or middle ear infection, and if the infection is left untreated, it may also progress to bacterial meningitis. Specimen that may be submitted for the diagnosis of strep pneumo may be sputum in cases of lobar pneumonia, or CSF in cases of meningitis, or blood if the physician thinks that the patient is now suffering from bacteremia. Characteristic appearance of the sputum samples for patients with lobar pneumonia has been described as rust-tinged sputum. Okay, medyo kulay kalawang na daw po, no? It's kind of a reddish-brown color. Okay, so again, that is referred to as the rust-tinged sputum, usually observed in cases of lobar pneumonia. Their colony morphology on BAP may vary, so may iba't ibang pwedeng maging itsura daw po ito on blood agar plate. Some are dome-shaped and mucoid, such as these ones right here. Okay, but most of them are characteristically umbilicate, meaning nakaangat yung gilid tapos nakalubog yung ilalim, just like these ones right here. Okay, so again, the correct description for this colony morphology is umbilicate. It's higher on the edges and lower in the middle. Okay, so again, that's an umbilicate colony, giving them a dimple shape or coin with raised rim appearance. Another description for strep pneumo is that they could be alpha hemolytic. One of its most important presumptive tests is taxo-P or optokin susceptibility. So if the isolate is strep pneumo, then it should be sensitive to the optokin disc and would have a zone of inhibition of somewhere that's equal or greater than 14 millimeters. So again, may pang-measure po tayo ng mga zones of inhibition na yan. Alright? So ayan po, no, it's quite easy to remember. Taxo P. Bakit taxo P? Because it's going to identify strep pneumo, 'di ba? So ayan po. Kanina nung pa-identify natin sa group A, Streptococci, taxo A, 'di ba? Which antimicrobial is the component of taxo A? Bacitracin. Okay, which species is going to be identified kung nag-taxo A ka? Streptococcus pyogenes. Alright? Pero dito, no, in this case, nasa strep pneumoniae na po tayo. So, taxo P ang gagamitin natin. Which antimicrobial is used in taxo P? Optokin. Another test that could help us identify if the isolate is strep pneumo is through the Neufeld-Kelung reaction. Basically, an anti-pneumococcal serum, which is a serum that contains antibodies to the pneumococci, is added to the sample and then it's examined microscopically. Okay, so if for example, may lobar pneumonia yung pasyente, so nagkolekta ng sputum. Yung sputum daw po, hahaluan natin ng anti-pneumococcal serum, which again is a serum that contains antibodies against the pneumococci, and a positive result is indicated by capsular swelling, as we can see right here. So kung sa microscopy po, nakikita nyo na may may mga capsules yung organism but you're not yet sure or you can't properly visualize them, you may proceed in performing the Neufeld-Kelung test. Alright? So kung strep pneumo nga po, yung isolate nyo, then their capsules should be enlarged or swollen. As we can see right here, mas na-emphasize or mas nag-enlarge yung capsules ng strep pneumo. So, ayan po, that's a positive result for the Neufeld-Kellang reaction. Here in Streptococci are constituents of the normal flora of the upper respiratory tract, female genital tract, and gastrointestinal tract. Okay, ulitin po natin, ang viridans strep ay normal flora ng mga lugar na yon. The term viridans means green, referring to the alpha hemolysis that many species could exhibit. However, a few beta hemolytic and non-hemolytic species have also been classified as virulent streptococci. Pero ayun nga po, no, most of them, they're capable of producing alpha hemolysis. So kaya ayun nga po, it leads to a greenish discoloration. So it was named viridans. So virulent streptococci are fastidious, and some of them require carbon dioxide for growth. They are divided into 5 groups, pero since hindi nga naman po sila ganun ka-clinically significant, and we usually just observe them as contaminants or normal flora, we will no longer be discussing these 5 groups in further detail. Okay, just remember yung mga possible diseases associated with them, such as for Streptococcus mutans. So as we can see right here, Streptococcus mutans could be found in the oral cavity and could lead to dental caries. All right, ulitin po natin yun. Ang Streptococcus mutans, it's part of the normal flora of our oral cavity. So what's happening here? What's wrong with that? Usually po, if a person doesn't properly perform oral hygiene like brushing their teeth, flossing, or mouthwash, no, there is a chance that these Streptococcus species could overgrow. All right? Maaari pong dumami sila, leading to tooth decay. All right? So ulitin po natin, Streptococcus mutans group could be a contributor in the development of dental caries. Okay? Take note, hindi lang naman mag-isa ng Streptococcus mutans group na nagdudulot ng tooth decay. All right? So it may be in combination with all the other normal flora of our oral cavity that could overgrow and eventually lead to the formation of dental caries. So ayan po, do not forget to brush your teeth 3 times a day. Enumerated here are some tests kung saan po pwede rin natin itong gamitin for the viridans streptococci. If we were to perform pyrrolidonyl arylamidase test, viridans strep would test negative. Dahil sino dapat ang magpa-positive dito? Streptococcus pyogenes. But on the other hand, we could perform the leucine aminopeptidase test. So sa leucine aminopeptidase po, dito magpa-positive ang viridans strep. Enterococci species consist of gram-positive cocci that are natural inhabitants of the intestinal tracts of both humans and animals. The commonly identified species in clinical specimens are Enterococcus faecalis and Enterococcus faecium. So Enterococcus sometimes exhibit a pseudo-catalase reaction or just weak bubbling, all right? Mahina lang yung effervescence that we're capable of observing in the catalase test kung ika-catalase nyo po ang Enterococcus species. All right? So let's have a review, no? Catalase test is usually performed to differentiate Staphylococcus from Streptococcus, 'di ba? And if you would remember, Staphylococcus species are catalase positive, 'di ba? And a positive result is indicated by effervescence or abundant bubble formation. So sinasabi po dito that Enterococcus species is capable of producing a weak result. Okay? So ayun po. Um, Identification of the species level is based on biochemical characteristics para daw po ma-differentiate natin yung Enterococcus faecalis and faecium. Now, in comparison, or if we take it in contrast to Streptococci, Enterococci have the ability to grow under extreme conditions. Okay, so tignan po natin yung physiology dito. For example, in the presence of bile or 6.5% sodium chloride, kaya pa ring mag-grow ng Enterococcus species. Okay, ulitin po natin, exposure to 6.5% sodium chloride, magso-survive pa rin yung Enterococcus. Is bile present in the environment? Magso-survive pa rin yung Enterococcus. Are we way beyond 35 to 37 degrees Celsius? Yung temperature ba kasing init na ng 45 degrees Celsius? Magsurvive pa rin ang Enterococcus species. Okay, so ayan po. Again, Enterococci are capable of surviving slightly harsh conditions, no, such as excessive salt concentration, presence of bile, or a much higher temperature. Okay, so enterococci are frequent causes of nosocomial infections. This would include UTI. So UTIs are most common, followed by bacteremia. So UTI is often associated with urinary catheterization or other urologic procedures. One of the most important presumptive tests in the identification of Enterococcus is the bile esculin test. So, other bacteria may be inhibited by salt concentration that is present in this culture media. Okay, so ulitin po natin, may mataas na salt concentration din po kasi yung culture media na ginagamit natin dito. But for Enterococcus, they will grow just fine. Okay? Enterococcus is also capable of growing in the presence of 4% bile. And they're also capable of hydrolyzing esculin to esculitin. Okay, ulitin natin, anong mga components ng culture media na ginagamit for bile esculin hydrolysis? Increased salt concentration, 4% bile, and esculin. Okay? So, ang Enterococcus species po, kaya niyang mag-survive in increased salt concentration, kaya niyang mag-survive with 4% bile, and kaya niya ring i-hydrolyze ang esculin para maging esculitin. Alright? So, the esculitin that was produced in this reaction, no, would then further react with the ferric ions that is present in the culture media. Okay, so may isa pang component ang bile esculin hydrolysis media. It has ferric ions. Kasi po, yung esculitin, kailangan niyang mag-react with the ferric ions so that we would be able to observe a dark brown or black precipitates, as we can see right here in this example. So blackening or a very dark brown appearance on the culture media indicates, no, the growth of Enterococcus species or a positive result in the bile esculin hydrolysis. All right? So again, ito po ay kayang gawin ng Enterococcus species na hindi kayang gawin ng Streptococcus species. On to the last segment of this discussion, we have the Streptococcus-like organisms. Itong mga susunod na organisms po, they're also not that medically significant, so we'll just go through them briefly and mga important points na lang po yung tatandaan natin. Biotrophia and Granulicatella species are formerly known as the nutritionally variant Streptococci. These bacteria grow as satellite colonies around other bacteria, and they require sulfhydryl compounds for growth. Okay, so ulitin po natin, they are formerly known as nutritionally variant Streptococci. So ano yung specific nutritional requirement na kailangan nila? Sulfhydryl groups. All right. So sulfhydryl, this is basically a sulfur bonded to a hydrogen atom. These organisms are part of the normal or the oral flora of humans, no, and their gastrointestinal tract. Okay, ulitin natin. A biotrophy and granulicatella could be part of the normal flora of our oral cavity and gastrointestinal tract. Next is Aerococcus. Aerococcus is a common airborne organism, pero pwede rin naman po itong ma-acquire through other forms of close contact. It is a widespread opportunistic pathogen causing an increasing number of infections associated with bacteremia, endocarditis, and UTI in immunocompromised patients. Aerococcus viridans has been linked to cases of bacteremia and endocarditis. Meanwhile, Aerococcus urinae and Aerococcus sanguinicola have been associated with invasive diseases such as sepsis, endocarditis, lymphadenitis, sorry, lymph not me literally choking on saliva. Um, lymphadenitis and peritonitis, which is the inflammation of the abdominal lining, often originating from the urinary tract. So UTIs are seen more often in elderly women. So Enterococci resemble virulent strep on culture but are microscopically similar to Staphylococci. Okay? So ulitin po natin, no? On culture, kung colony morphology ang titignan, ang kamukha daw po ng Enterococcus species ay yung virulent strep. But microscopically, if we were to look at them, ang kamukha daw po ng Enterococcus species ay ang Staphylococcus species wherein they could occur as tetrads or clusters as we can see right here in this photo. Kapag sinabi po na tetrads, they're arranged in this kind of like 4 by 4 cube shape. All right, so that's for Aerococcus. Next is Dolocycoccus and Globicatella. So for Dolocycoccus persevorin, this is actually the only species in this genus. Okay, so mag-isa niya lang na Dolocycoccus. And sometimes this could be isolated from blood samples. Next, for Globicatella species, we have right here Globicatella sanguinis. All right, so this is the only species capable of causing human infections. All right, so ulitin natin, there may be other Globicatella species. It's just that Globicatella sanguinis, ito lang yung na-associate with human infections. So diseases associated would include bacteremia, UTI, meningitis. No? So listed here are the samples that we could collect in cases of these types of infections. Next is Gamella. Gamella species are similar in colony morphology and habitat to the virulent Streptococci. Okay, ulitin po natin, no? Magkaparehas daw po yung usual habitat and colony morphology ng virulent Streptococci. So, they produce alpha hemolysis or some of them could be non-hemolytic. So, Gamella could easily decolorize on gram staining and often appears as gram-negative cocci in pairs, tetrads, or clusters. Alright? So, sobrang bilis daw po na decolorize ng gemella na minsan they might look gram-negative kahit gram-positive naman talaga sila. Okay? So, Gamella species have been isolated from cases of endocarditis, wounds, and abscesses. The most significant species is Gamella hemolycens. Lactococcus species are gram-positive cocci that occur singly or in pairs or in chains and are physiologically similar to Enterococci. Kapag sinabi nating physiologically similar to Enterococci, maaari pong may mga test results, no, na parehas ang result na kayang i-produce ng Lactococcus and Enterococci. Now, on SBA, these organisms produce alpha hemolysis and are Ayun po, alpha hemolysis or some of them may also be non-hemolytic. Next is Leuconostoc. The genus Leuconostoc consists of catalase-negative gram-positive microorganisms with an irregular crocoid morphology. So these organisms share several phenotypic and biochemical characteristics with Lactobacillus species, virulent Streptococci, Pediococcus species, and Enterococcus species and sometimes are misidentified. Okay? So ulitin po natin, although rare naman na ma-isolate in the laboratory itong mga organisms na to, mahalaga po na aware pa rin tayo sa kanila kasi sometimes we could misidentify them as the major pathogens that we've discussed earlier. Okay, ulitin po natin, some of these streptococcus-like organisms may easily be mistaken as the actual pathogens that we've previously discussed. So we also have to be aware of their presence. All right, so ulitin natin, Leuconostoc species, they could easily be misidentified with Lactobacillus, Viridans strep, Pediococcus, and Enterococcus species. Okay. Next, members naman po of Pediococcus are facultatively anaerobic and they are also gram-positive cocci that could vary in arrangement. Paiba-iba daw po ang possible arrangement ng Pediococcus. Okay, they could be in pairs, they could be in tetrads, they could be in clusters, and they could grow at 45 degrees Celsius. Which organism ang na-discuss natin kanina na kaya ring mag-grow at 45 degrees Celsius? Enterococcus species, di ba po? So for pediococcus, they may be misidentified as either the viridin streptococci or enterococci. Here are the references that were used in the making of this discussion. That ends our discussion on chapter 15 of Mahon's textbook of Diagnostic Microbiology. This has been Aurel Enriquez. Thank you so much for watching.